Hello Year 12s, hope you're all okay and safe. Obviously unprecedented times like these call for different ways of teaching and learning. So what I've done is I am creating a video to show the first stage of photosynthesis, which we call the light dependent stage. So first of all, we've got to assume that we are in the chloroplast of a plant cell. And we are looking at a membrane of the thylakoid. So this is the thylakoid. Membrane. And then we have the stroma, which is the fluid of the chloroplast. And then we've got the thylakoid space, which is the internal part. Okay, so if you imagine that we are in the chloroplast and we are actually looking at one of these lines here, the membrane of these stacks of thylakoids, which we call grana, remember. Okay, so we're looking at the first stage of photosynthesis, which we call the light dependent stage. So let's go back to this. Within the membrane, we have two proteins, okay, and these are called photosystems. So PS denotes photosystems, and we have photosystem 2, and we have photosystem 1. Photosystem 2 being discovered after photosystem 1, but actually is used first in the light-dependent stage of photosynthesis. Each of these photosystems absorbs light, and they absorb light at different frequencies or wavelengths, and therefore this one absorbs light at P680 nanometers, and this one at P700 nanometers. Really important that you remember the wavelengths in which they absorb that light. So, plants take in water, and they use that water within the chloroplast. They also take in light. So let's have our light energy up here. And what's going to happen is we're going to go through a process called photolysis. Lysis meaning to split, photon from light. So what's going to happen is this wave of light is going to come down and into the photosystem. Now the photosystem has chlorophyll A, chlorophyll B and carotene, which are the photosynthetic pigments in which the plant needs to be able to photosynthesize. So photolysis happens and water is split into two protons or 2H plus and half an O2. The two electrons as well are in the photosystem, and they become excited as a result of that light energy. Now, the exciting of electrons we call photoionization. So we've got photolysis, splitting of water, and we've got photoionization, the excitement of electrons in the photosystem. Now, whenever you see diagrams, the height within the membrane denotes the amount of energy they have. So you can see that when the electron is down here, it has very little energy. As it is excited through photoionization of light, it gains energy and so moves to the top of the membrane. Between photosystem 2 and photosystem 1, we have electron carriers. That's going to transfer the electron from photosystem 2 to photosystem 1. Now, we have a high concentration of protons outside in the stroma. So this electron has gained energy and moves to the top of photosystem 2. As it's no longer being excited by this light ray or photon, it loses its energy as it passes from photosystem 2 to photosystem 1. And you can see that it moves gradually down to the bottom of the membrane, so therefore it loses energy. When it loses this energy, that energy is used to pull through the protons from the stroma into the thylakoid space, H+. And we call that chemiosmosis. So this chemiosmosis, just think osmosis, the movement of molecules from a high concentration to a low concentration, and it's chemi, so therefore it's chemicals or ions. Now, because you have so many of these electrons moving through, you build up a high concentration of protons here and a low concentration of protons out here. We've got this protein here. Now this is a protein and this is an enzyme, a membrane-bound enzyme called ATP synthase. Okay, so ATP synthase, just think synthesizing ATP. So because we build up a large concentration of protons, because we've got continually lots of electrons moving through, we're going to have diffusion of these protons from high concentration to low concentration back out. Now, as protons move back out, the ATP synthase is going to synthesize ATP. So as it moves out, the head of the ATP, this part here, spins and it joins ADP and inorganic phosphate to make ATP. So we've used light effectively to make ATP. That is called photophosphorylation. Okay, so that process is called photophosphorylation. The phosphorylation, the adding of phosphate essentially, so phosphate to a molecule, ADP, and photo, we're using light, so we use light to add phosphate to ADP. Now, let's go back to photosystem 1. We have our electron now in photosystem 1, and photosystem 1, just like photosystem 2, is going to absorb light, but this time at a slightly different wavelength, 700 nanometers. And so light is going to come in to photosystem 1, and again we're going to have photoionization of this electron, but this time it's going to leave the photosystem. We have a molecule called NADP, and that is going to join with the H+ that's come out through the ATP synthase. So we're going to join those two together, and that is going to form NADPH, or reduced NADP. Okay, you can either write reduced NADP, or we can write NADPH. Now this electron is also going to join that reaction, because if we add an electron to a proton, we make a hydrogen atom. So this NADPH and this ATP is then going to go to the second stage of photosynthesis, called the light independent stage, or the Calvin cycle. Okay, there is no light needed there, so hence why plants can still photosynthesize at night time. And that is pretty much it, okay, for the light independent stage. So, just repeating, we have the thylakoid membrane, we have two photosystems, photosystem 2, photosystem 1. Each photosystem absorbs light at a different wavelength, 680 nanometers, 700 nanometers. And we have photolysis of water, because light comes in, splits water into protons and oxygen. The two electrons become excited through this in a process called photoionization, and they move up the photosystem and then move across electron carriers, and as they do that they lose energy, and that pulls in protons through chemiosmosis. These protons then move out through the enzyme ATP synthase, and as it does that, it photophosphorylates ADP and PI to turn it into ADP. This proton can then combine with NADP when the electron is re-photoionized or photoionized for the second time in photosystem 2. Our NADP and our ATP are essential for the light-independent stage, or the Calvin cycle. Okay? Without NADPH and ATP, you cannot go through stage 2. So the whole point of the first stage, or the light-dependent stage of photosynthesis, is generating NADPH and ATP. Hopefully that makes sense. If you have any questions, just shout. Thank you very much for listening. Bye-bye.